This is Dr. Double B, and this is a shorter lecture on homologous recombination deficiency. So just to show some H&Es for fun, this is particularly important in ovarian cancers, high-grade serous, prostate cancer, breast cancer, and pancreatic cancer. So homologous recombination deficiency refers to defects in the homologous recombination repair pathway in these genes and proteins. So here we have a double-strand break, and it's supposed to be recognized by different proteins like ATM, ATR. It's prepared for repair by other proteins like the RAD50, Nibrin, MRE11. And then other ones will actually oversee the repair like BRCA2, RAD51, and there's a number of more proteins in this pathway. And so it's called homologous recombination repair because, going backwards a little bit, it's using this homologous chromosome as a template for the repair. And so what you'll also have is a loss of heterozygosity in this area. So anything that was different between the two chromosomes will now be the same. And there's slightly different ways that this can happen, but I animated one of the ones that I thought was straightforward. So these are some of the genes involved. A subset of these will be important for testing, but then these approvals and things could always be expanded in the future. So hopefully some of these look familiar: BRCA1, BAP1, BRCA-associated proteins, and some of these others. So there's another pathway that's kind of distinct but kind of related as far as treatment, and that's the PARP pathway. So PARP stands for poly-ADP ribose polymerase. So this is involved in single-strand repair. And so this one, you can imagine, is a little bit simpler. You have the other strand to use as a template, so you can basically just fill in the missing nucleotides here. So this should be error-free and not really lead to any changes. And so they're connected because single-strand breaks can become double-strand breaks. So say you have a single-strand break and the cell is dividing, strands are replicating. So that single-strand break is now going to become a double-strand break that would need to be repaired by the HRR, homologous recombination repair pathway. So again, HRR proteins repair double-strand breaks, PARP repairs single-strand breaks. And so this was a creative idea called synthetic lethality. So historically, a lot of the cancer drugs are inhibitors of oncogenes or oncoproteins. So something is overactive like BRAF, EGFR, KRAS, something like that. So you develop an inhibitor for it. It's been harder to target tumor suppressors because there's not a lot of ways to reactivate a tumor suppressor. But there's an alternative, which is to kind of find the cells that have a defect in the tumor suppressor and then give something else to kind of kill them off. And so that's what's done here. So say you have a normal cell and a tumor cell, tumor population that's HR deficient or HRD. Now they're going to randomly have single-strand breaks, and they're also going to have these as a side effect of some of the chemos that are given in these tumors as well that are going to increase the single-strand breaks. And then say you give the patient a PARP inhibitor, those single-strand breaks will not be able to repair, and when the cells divide, they're going to become double-strand breaks in the normal cells and in the tumor. But now the normal cell still has functioning HRR pathways, so it's going to be able to repair those. The tumor cells, on the other hand, can't repair those, and over time, they're going to have too much DNA damage, and it's going to lead to cell death in the tumor cells selectively. So these PARP inhibitors have names like olaparib, talazoparib, and again the idea is the cells without BRCA or other HRR pathway proteins are going to die off. And there are small molecule inhibitors like that. So here's an animation I borrowed from YouTube. So we have a single strand break, and we have the PARP protein here. So the PARP protein would normally recognize that and repair it, but the drugs, there's a trade name, Lynparza, is going to bind and prevent it from repairing and actually trap it there on the DNA. then the single strand break is going to become, again, a double strand break that now in the tumor cells can't be repaired by that HRR pathway. And so one of the main indications for giving these drugs is loss of function proteins in those HRR genes that showed at the beginning. So if you have a BRCA mutation, then they're going to have this defect, and you can treat with the PARP inhibitors. But there was another approach, and instead of looking for specific mutations, there's additional genomic evidence of HRD, and this is referred to as genomic scars. So we have scarred characters here. And so a lot of those rely on looking for LOH, or loss of heterozygosity. So in normal case, when you have homologous chromosomes, you should have some variety between the two. So you have these single nucleotide polymorphism sites that vary randomly between people and between chromosomes. So some of them will be the same. You might have a C on both, and some will vary. You might have different nucleotides on different ones. And these are just generally normal and just contribute to variability between people. Again, between your chromosomes, you're going to have different ones as well. And something like 0.1% of nucleotides that normally vary. And of course, the exact percentages will vary by the site. And so in the case of a loss of heterozygosity, this is referred to as a copy neutral one because a deletion would also be a loss of heterozygosity if you just deleted it. But in this case, this is copy neutral, so one has basically been deleted and the other copied over here. So now you no longer have these heterozygous ones where you have an A and a T, a G and an A. Now everything is just the same between the two chromosomes in this region. And the reason this is important is this is one of the main genomic scars. So say you have a double-strand break here, and the cell is dividing, so you have your homologous chromosomes and sister chromatids. So they can actually recombine in the area of this double-strand break. And so then we continue with the cell division. And now in the subsequent cells, you have these regions of loss of heterozygosity. And in this case, it happens to be near the telomere. So on this one, you have all of the light blue version. On this one, you have all of the dark blue version in this region. Okay, so showing this again, this is a loss of heterozygosity, and there's thresholds for a lot of these. So for it to count as a genomic scar when we do this HRD score, it has to be at least 15 megabases. And so there are three components. The first is just the LOH. The second one is called telomeric allelic imbalance, which is LOH or another CNV at the telomere of at least 10 megabases. So this one change actually qualifies as both of these, an LOH and a TAI. And then there's one more thing, which is a large-scale state transition, which is any change in allelic content. So like LOH and deletions and amplifications of at least 10 megabases. So this one change actually qualifies as all three of these things. So it would add 3 to your score. So again, this is a little bit technical, but the HRD scores the sum of these three things. And there are the definitions one more time. LOH, loss of a parental allele, loss of one of your alleles via LOH. So copy neutral or loss deletion. And then TAI, these regions, variable regions at a telomere and not crossing the centromere. And then LST, large-scale state transition, are these any changes. So when it changes over to a different kind of allelic content where there's a deletion, duplication, LOH, that'll count as 1. And so again, if you're counting this up, it would be over all of the chromosomes, over all of these changes. But one change can count as multiple, so this alone counts as 3 towards our HRD score. And then here's a graphic I borrowed. Fortunately, I remember the reference here. It shows some of these. I don't want to go through all of them because it would take too long. But again, this is showing kind of these changes in allelic content, LOH, etc. So the first one, they're showing that this one was basically copied over here. So now you have LOH, it's at the telomere. There's one LST here, so that gives you 3. That's basically the one I just showed. This is a deletion. So again, it counts as all 3 because it's an LOH. You have loss of heterozygosity, but from that deletion, you have an LST here, and then you have, it's at the telomere, so it's a TAI. And then Here's another one, kind of similar, but it doesn't include the telomere, so that one doesn't count. They're kind of using HRD just for the LOH here. This one, there's not really the LOH event, but there is a change in content of the telomere, and there's a position where it transfers over, so it's an LST. And so on. You can kind of go through these if you're really interested. Here's some graphics, I again borrowed from a lecture from the faculty at University of Hawaii who are doing some work and showing some of these examples. This is how you would look at it in an array. I think I'll do a separate lecture on arrays, another shorter one. But basically the dots are kind of showing you where you have a change in content, like a deletion. So here was one here, so you'd have this LST here and here, and LOH in this region. Looks like a deletion. And then here's another one kind of occurring near the telomere here. So again, LST break, and then near the telomere, an entire region here. And here was one also borrowed from another lecture. I'll get some of my own eventually in the near future, actually, I think. But here we're showing some negative cases, low positive and high positive. So this is showing over everything. It's a little small, but you can kind of see the different chromosomes are just lined up here, one after the other. And so sometimes you'll see some normal variations, particularly at, around telomeres and maybe centromeres. So you'll have some in the background. So this was a negative case. And then here's a low positive case where you see a lot more of this variability, changes in allelic content. Again, it's hard to tell exactly where everything is on this because we're zoomed super far out. And then a high positive where you have a lot of variation. And so you'd imagine a lot of these things would qualify as all the things we talked about, LOH, TAI, and LST. Again, I think I'll do another lecture focusing a little more on how to read arrays. All right, and so here is, I think, the initial study that kind of set some of this up. And they're looking at the HRD score in cases that are BRCA intact in red and BRCA deficient in blue. And so there's kind of a threshold here of 42 where it passed over from most of the negatives to most of the positives. And you notice there is a little bit of an extra peak over in the red, so that's probably things that are caused by defects in other genes other than BRCA. So the approvals get a little bit complicated here. It depends on the tumor type. So like ovary, these can also change. Somatic or germline defects in BRCA can get the PARP inhibitors or the high HRD score. That's particularly high-grade serous carcinomas. And then breast and pancreas, germline defects in BRCA. And then prostate, somatic or germline defects in a set of the HRR genes that have that FDA approval. So it's a little complicated to keep track of. It's kind of complicated in general, I guess. But that's all for now, and stay tuned for more.